In a major leap forward for diabetes research, scientists at the University of Geneva have uncovered a potential game changer in the form of a protein called S100A9. Unlike traditional therapies that rely on insulin, this naturally occurring protein appears capable of regulating dangerous blood ketone levels and stabilizing blood sugar without the risks typically associated with insulin. The implications of this discovery are not only innovative but genuinely hopeful for millions of people around the world living with type 1 diabetes or severe insulin deficiencies. For over a century, insulin therapy has been the standard treatment for diabetes. While it has been life-saving, it also carries its own set of complications: hypoglycemia, difficulty in precise dosing, and long-term metabolic risks among them. Daily injections, constant monitoring, and the ever-present fear of dangerous swings in blood sugar create a heavy physical and emotional toll. The University of Geneva team has now identified an alternative that may reduce, or even eliminate, that burden in some cases. Their discovery centers on how the body manages ketones, which are energy molecules produced in the liver during periods of fasting or insulin deficiency. While ketones are a useful backup fuel source under normal conditions, they can become dangerous in excess, particularly for people with type 1 diabetes. The condition, known as diabetic ketoacidosis, DKA, is life-threatening and leads to thousands of emergency room visits each year. Until now, only insulin could effectively shut down this runaway ketone production. But in this new research, the protein S100A9 demonstrated the ability to do the same safely and effectively. This is not just an academic discovery. In experiments with diabetic mice, S100A9 did more than regulate ketones. It also improved blood sugar and lipid profiles and extended the lives of insulin-deficient mice without any harmful side effects. It achieved this by activating a receptor on liver cells known as toll-like receptor 4, TLR4, setting off a beneficial chain reaction inside the liver that ultimately reduced ketone production. Surprisingly, this same receptor, often associated with inflammation, did not trigger any inflammatory response when activated by S100A9. Instead, it appeared to have an anti-inflammatory effect. That's a significant bonus. The team's next step is to move from animal models to clinical trials in humans. Thanks to a generous 2.5 million Swiss francs grant from the Helmsley Charitable Trust, this transition is now within reach. That funding will complete the remaining preclinical studies and begin the process of applying for regulatory approval. If those trials are successful, S100A9 could become a widely used injectable treatment that either complements or, in some cases, replaces insulin, offering a new layer of safety and flexibility for diabetes management. What makes this even more exciting is the potential for broader applications. Because the S100A9 pathway does not provoke inflammation, it might be useful for other diseases involving poor metabolic control or chronic inflammation. That opens a door to treating not only diabetes, but also a range of other health conditions with fewer side effects and a more targeted approach. This breakthrough also signals a shift in the way researchers are thinking about diabetes therapy. Rather than simply trying to improve insulin or reduce the amount needed, S100A9 shows us that it's possible to find an entirely new route, a therapy that works independently of insulin, possibly bypassing many of the complications that have plagued diabetes care for decades. The future of diabetes treatment may no longer hinge solely on insulin. With the discovery of S100A9, scientists are bringing us closer to a world where people living with diabetes can manage their condition with less risk, greater comfort, and more optimism. The development of a real-world therapy based on this protein is now well underway, thanks to Diathris, a new startup launched by the researchers. Supported by the University of Geneva's innovation ecosystem, they are pushing forward with the goal of making S100A9 a safe, effective, and accessible treatment. In a world where chronic illnesses often mean a lifetime of compromise, this development brings not just scientific progress, but real hope. The path ahead will still require rigorous testing, regulatory review, and continued innovation, but it's a path that now feels full of promise.